So this is our section B remediation for the prelim, okay? So this is question 1A. Now, what it's asking us in our question here, right, is it's just saying to us, choose one of the structures labelled and state their functions. So if I pick cell membrane, what does it do? It controls entry and exit of molecules to the cell. Okay, or it allows entry and exit of molecules to the cell, or it allows entry and exit of small molecules to the cell. Okay, cytoplasm, site of chemical reactions. Ribosome, site of protein synthesis. And mitochondria, site of aerobic respiration. Okay? So the next question, 1B here. The field of view of a light microscope measures 2 millimeters in diameter. So usually we see this as a wee picture and it tells us, okay, the diameter of this is 2 millimeters. Okay? Now the answer, look at this, the answer is in micrometers. So what I want to do is 2 millimeters. I want to change that into micrometers. Now look at this bit here. It says 1 millimeter is 1,000 micrometers. So I've got 2 of those, so 2,000 micrometers. Okay? So across the way, what I've got here is I have got 2,000 micrometers. That's my size. Okay? Now it's not given us a picture to count the cells across, but look at what it's done. Is it's told us how many cells are going across. So I do 2,000 divided by 20, and that gives me 100. So this wee experiment, right, this is question 2. The apparatus shown below was used to investigate the movement of water. So look at this. It's telling us it's the movement of water. So what we've got to think there is, okay, osmosis is happening. It says into and out of the model cell. The model cell had a selectively permeable membrane. Okay, so what I've got here is water outside and 10% salt solution inside. Now, the first thing I would always do is draw on my picture and decide where is the high water concentration, where is the low water concentration. So the water or the salt, what one's got more water? The water. High water concentration. Lower water concentration, we say the definition of osmosis to ourselves. The movement of water from a higher water concentration to a lower water concentration through the semi-permeable membrane. So we know that water is going to be moving in, okay? Now what the question tells us is the liquid level in the gas tubing was measured every 10 minutes for 60 minutes, and the results are shown in the table. So we can see there that the water's moving in, because look, the liquid is rising because the water's moving in, okay? Name the process which caused the liquid level to rise. Osmosis. So that's our very first mark here. Next question. So look at this, B. It says explain how this process caused the liquid level to rise. Now, the first thing that I said that we would do is decide where's the high, where's the low. So water's got the high, the low's inside, which means water moves in. So by drawing on this picture, we've answered this question. What do we say? Water moves from a higher water concentration. Now, where is that higher water concentration in the picture? Outside. To a lower water concentration. Inside. And I go through the semi-permeable membrane. Okay, now that answer would get me 2 marks, okay? However, the other thing that I could do is just give the definition of osmosis first. So I could say, okay, the water moves from a higher water concentration to a lower water concentration through the selectively permeable membrane. That would give me 1 mark for the definition of osmosis. And I could then just say the water moves in, and that would get me my 2nd mark, okay? Now let's look at the next question. So C, calculate the average rate of movement of liquid in the gas tubing, right? And what it's saying down here is that wants it millimeter per meter. Millimeter per minute, okay? So what we've got to think is, we've got to think about our results, right? So we've got to get our wee table of results up. Okay, so here's my table of results here. And remember, we're looking for millimetres per minute. So have a look. We have gone 60 minutes, okay? And how far has the liquid moved? It's moved from 10 to 64. So how far has that moved? It's moved 54 millimetres. So I've moved 54 millimetres in 60 minutes, but what do I want it for? 1 minute, okay? So if it's moved 54 in 60 minutes, then for 1 minute, I do 54 divided by 60, and it gives me 0.9. So question 3. The diagram below shows stage in a production of a protein. We've got our wee picture here. Here's the ribosome, here's the amino acids, and we have this molecule Y. The above shows an example of protein being synthesized. Name molecule Y and describe how it determines the sequence of amino acids as shown. So look at this molecule Y. Who is he? Look, he's one strand. He's at the ribosome. He is messenger RNA. Okay? So molecule Y, messenger RNA. Now, how does it determine the sequence of amino acids? There's a couple of answers here. I could literally just say the sequence of bases, because it's the sequence of bases that determines the sequence of amino acids, right? Or, because this is mRNA, what I could do is, well, what does that do? It takes a complementary copy of the DNA from the nucleus to the ribosome. Because by taking that complementary copy of the DNA from the nucleus to the ribosome, we're taking the sequence of bases, and those sequence of bases will determine the sequence of amino acids. Look at part 2 down at the bottom. Name the part of the cell where the protein was made. Ribosome, site of protein synthesis. So this was us on to question 4 now. Okay, so question 4, it says the diagram below shows some stages in cell division. Okay, we've got our wee pictures, and the first question is, what name is given to this process of cell division? Mitosis. Now the next question is asking us to state what's happening in stage D and C. So let's look at C to begin with. Okay, so look at this. I've got this little dude over here and this little dude over there. And look at my wee spindle fibres. Here's my wee spindle fibres. What's happening there? Chromatids are pulled apart to opposite sides of the cell, right? Instead of pulled apart, I can say separated. Okay, so the chromatids are pulled apart to opposite ends of the cell. The chromatids are separated to either end of the cell. Look at stage D. What's happening in stage D? Well, look at this. Look at what's happened. New nuclear membrane is formed. So I will get a mark for that. But also, look at what's happening at this bit. Cytoplasm divides. Either one of those I will get a mark for. Last question down the bottom here. Why is it important that the daughter cells receive the same chromosome complement as the original cell? So that they can function correctly, so that they can function properly, so that they can carry out the same function as the original cell. Question 5. Respiration in cells is the process in which energy is released from food. The diagram below shows stages of respiration in an animal cell. So what we've got here in this picture is stage 1, glucose to substance X, and then we've got this stage 2, CO2 and water. Describe the process of aerobic respiration. So what I want to do here is talk through both stages, okay? So my stage 1, what is this? It's glycolysis, okay? So what we want to then think is, what's everything that I know about glycolysis, right? So it happens in the cytoplasm. What do I not need? Don't need any oxygen. What am I breaking down? Glucose. So glucose broken down into pyruvate. What else do I get? 2 ATP is made. Okay, stage 2. That's my aerobic stage. So we think aerobic stage, where does this happen? In the mitochondria. What do I need? Oxygen is required. Now, I've already named that this is pyruvate, this substance X. So if I've already said that the glucose was broken down into pyruvate, then I don't then need to say, oh, the pyruvate's broken down into CO2 and water, because look, it's in the picture, okay? So what else do I get? I get 36 ATP produced. And I get 38 ATP produced in total for 1 glucose. Last thing that I could say is the whole process is enzyme controlled. Okay. Now essentially what will happen is this is a tick, this is a tick, this is a tick, this is a tick, so is this, this is, and then these two, you can have one or the other. And then enzyme controlled would be a tick. Every 2 ticks gives you 1 mark. Okay, so if you get like 3 ticks, it's still only 1 mark. There are no half marks. Next question. So in the absence of oxygen, substance X cannot be converted to CO2 and water, and an alternative anaerobic pathway occurs. Now, our anaerobic, that is fermentation. And see in your exam paper, it will say fermentation instead of anaerobic respiration. So state the products of fermentation in yeast. 2 things. I get alcohol, or I can call that ethanol, and CO2. I must have both answers, okay? Now look at the next question. Give an example of a cellular activity that uses energy released in respiration. So something that I need energy for. Active transport. However, there's lots of answers here. Protein synthesis. I could say mitosis. DNA replication. Muscle contraction. Lots of options there. Okay, so why does a sperm cell contain more mitochondria than a skin cell? So what we've got to think is, what does the sperm cell do that I would need more mitochondria? Well, what does it do? It swims. So why am I needing more mitochondria? It needs more energy to swim. Where does it swim to? The egg. Okay, so question number 6, right? What we've got here is a wee enzyme experiment. Now, the first question it's asking for is for us to just draw the graph, okay? So what you can see here is the volume of gas produced. So look at that there. That means that my temperature has to go along the bottom here. So the very first thing I'm going to do is write my label, temperature, okay? Now, look at this heading. What else has it got? Degrees C. I must have the degrees C, okay? Now, I go up from 10 to 50. Look, it's already got the 0 here. So I just want to look at it and go, can I go 10, 20, 30, 40, 50? Yeah, I can. So 10, 20, 30, 40, 50. Okay, I need to make sure that it's a scale. I need to make sure that each box is worth the same thing. So I've got 10 wee boxes going up to 10. Each box is worth 1. Okay, up the side here, we can look at how much is each box worth. So look, I've got from 0 to 5. And see 5, it's 10 boxes that's taken me up to that. So 10 divided by 5 equals 0.5. That's 1 box on the way up is 0.5. Okay, so all I now have to do is plot my points. So the very first point I'm plotting is 10 degrees C. And I want it to be at 5. So there's my 10 degrees C, and here's my 5 for the volume of gas. 20 degrees C, I want 12, so 20, I go up to 10. Now I have to remember that each box is not worth 1, each box is worth 0.5, so I need to go up 2 boxes, that's 11, and then another 2 boxes, so here's my 10, 1, 2, that would be 11, oh, sorry, that would be 11, and another 2, sorry, wee box keeps popping up, so that's my next point there, okay. 30, I want to do 14. So again here, I got up to 10, but I need to get up every 2 boxes as 1. So where's my 14? Here. 40, I've got 19, so my 19 is going to be here, and then the last one is 15 right on this line, okay? Now, what we now need to do is we need to get a ruler. Now, I might not be neat with this because I can't actually physically use a ruler. So what you do is you get a ruler and you just line up and you join a straight line dot to dot, okay? And again, your line would be neater than mine because I'm not able to use a ruler, okay? So that's my line. 2 marks for your graph. 1 for the label and this scale along the bottom, and 1 for plotting my points and drawing my line. So this next question, it's saying calculate the percentage increase in volume of gas produced when the temperature rises from 10 to 40. So volume of gas at 10 is 5, volume of gas at 40 is 19. So percentage increase, I use the equation percentage change, is the change divided by the start, and then what I do is I times this by 100. Okay, so I look at these two numbers. What's the change? The difference between 19 and 5 is 14. Okay, I now have to divide that by what it started as. What did it start as? 5. And then times by 100. When I pop that into my calculator, I get 280% increase. Okay, what you've got to do is you've just got to know this equation. Percentage increase, percentage decrease, it's a percentage change. So it's the change divided by the start times by 100. So again, still this enzyme question. It's saying predict the volume of gas which would be produced if the investigation was repeated at 60. What's going to happen to my enzyme at 60? It's denatured. Now I never say denatured by itself. What does denatured mean? The active site changes shape. Okay, so if the enzyme's denatured and the active site changes shape, what's it going to be? Nothing. Right, it's not going to happen. Bottom question here. The procedure above could be adapted to investigate a variable other than temperature. So what you've got to do is look at this. When I was changing temperature, what have I got? I've got a range of different temperatures, right? So I want to change this so that I'm not investigating temperature and I'm investigating something else, right? So any one of the 3 of these that I pick, right, what am I going to use? A range of different, and whatever it is I'm picking, so a range of different concentrations of enzyme, a range of different concentrations of substrate, a range of different pHs. Now, if I've got a range of different one of these, what do I have to do to temperature? I'm going to have to keep temperature the same. I'm going to have to keep the temperature constant, okay? Now, if we just look at how this question was actually set out. So look at how it's set out here. There's my variables. And then it said, chosen variable, adaptation 1, adaptation 2. Well, adaptation 1 is my range of different, and then whichever one of those I picked, right? So like pH, for example. And then temperature, keep the same. And that's us. Question 7. So here it's giving you a graph and it's giving you some chat that you know nothing about. So it's saying cancer is an example of uncontrolled cell division. Melanoma is a type of skin cancer caused by the overexposure to UV light from the sun. The graph below shows the incidence of melanoma in men and women over a 30-year period. So I've got year along the bottom, and then I've got women, I've got men, 2 different lines, and then up the side here I've got cases per 100,000 of the population. Using information from the graph above, give 2 conclusions. Now, see this, what you've got to think is, if I'm not looking at the graph, you need to give me 2 conclusions that's going to allow me to picture this graph, right? So the first thing you want to think is, all right, what's along the bottom and what's happening up the side? So as the years are increasing, what's happening to the cases of melanoma, right? So as years go by, go on, right, whatever, cases of melanoma increase. They're rising. They're going up, right? So I then know that I've got a graph and my lines are going up, okay? But again, there's 2 lines on it. So I need to know, well, where are these 2 lines in relation to each other, right? So what's the difference between the males and females? Females have more melanoma. And that's my second conclusion there. If I say that as the years pass, the cases of melanoma are increasing, then I know that this line is going up the way like this. If I say more females have it than males, then look at this, females, males. I get that gist as well. Okay. Question 8. The diagram below shows a process that occurs in the reproductive organs of mammals. So first things first is just asking for process X. So look at this. What is process X? Fertilization, because look at that, the sperm and egg are coming together. What's the cell made in fertilization? So cell Y, zygote. What is meant by diploid? The cell has 2 sets of chromosomes. I think of the di as being like for double, right? So I think di stands for double. And then I think the ha in haploid stands for half. So diploid have got 2 sets, haploid have got 1 set. Question 9. One type of deafness in humans is caused by a single gene. The diagram below shows the pattern of inheritance in one family. A capital H represents the hearing form of the gene. So that hearing form of the gene, right, is a capital H. Hearing form of the gene, capital H, okay? Little h represents the non-hearing form of the gene. So non-hearing is my little h, okay? So look at this. John, it's telling us, has a big H and a little h, and it's telling us Linda has little h and little h. Now, I can fill in some people already here. Look at Paul. Paul has to be 2 little h's. Has to be, can't be anything else, right? Because he can't hear, so he has to have 2 of the recessives. Look at Jackie, right? Jackie can hear, so she's got to have a big H. But what did Paul give her? Paul gave her a little h. Lyle, he can hear, so he's got a big H. But then look again, what did Paul give? Paul's his dad, look. Paul gave a little h. Okay. Now, very first question here is saying, using John as an example, explain how it is known that the hearing form of the gene is dominant. So what we've got to think here is, all right, well, what's John? John has a big H and a little h. How do I know that it's the big H that's dominant? He can hear. That's it. He has got a big H and a little h, he's heterozygous, and he can hear. Part 2. Use information in the family trees to complete the following table to show the genotype and phenotype of each individual. Now look at this, we've actually already done it. So Paul, he can't hear, so he's got 2 little h's. The phenotype, non-hearing. Lyle, look at Lyle. Lyle can hear, so he's got a big H, but then again, look, Paul's his dad, so Paul gave him a little h. And what's his phenotype? Hearing. Okay. Now, what we're doing now is it's saying Fiona, okay? So let's look at Fiona here, up in the family tree, okay? Fiona can hear, so she's got a big H. But look at her mum, look at Linda. Linda's given her a little h. So Fiona is big H, little h, okay? She has a child with a man who's the same genotype as her, big H, little h. And we want to find out what's the chance of their child being able to hear. So we draw our Punnett square. Big H, little h, big H, little h. So that gives us 2 big H's, a big H and a little h, a big H and a little h, and then a little h and a little h. What is the chance that their child will be able to hear? Look at this. 1, 2, 3. It's a 75% chance. Or it's a 3 in 4 chance, right? We don't want to just do the ratio because 3 to 1, that's not actually, if I just write down 3 to 1, it's not actually really telling us. What's the chance? 3 in 4, 75%. So we problem solving question here. First bit it tells us is this little bit of chit chat at the top here. Stem cells are unspecialised cells which can divide to make a wide range of other cell types in the body. The embryo contains the highest proportion of stem cells. The chart shows some of the specialised cells that are formed from stem cells. So look here, I've got unspecialised cells. That's my stem cell, look. I've got these intermediate cells. So the intermediate cells, that's this basic bone, young fat cell, primitive blood cell, pre-cartilage cell. And then we've got this mature, and here's my list of mature underneath that. So here's all my mature cells, okay? First question it's asking us is which intermediate cell type can develop into the widest range? So here's my intermediate cell. Basic bone, how many cells can that turn into? 2 different cells. Young fat, only 1. Primitive, there's 3 different. Pre-cartilage, there's only 2. So which one of those can develop into the widest range? The primitive blood cell. Next question. Which type of intermediate cell forms both elastic and non-elastic fibres? So all I've got to do here is find the chat that's talking about elastic and non-elastic. So look at this. Elastic here, non-elastic there. So who is this? Pre-cartilage cell. So there's my answer there. Pre-cartilage cell. Okay. Now, I'm just going to go to my next slide so I can see a little bit more. So see this here. What I wanted to do here is I'm keeping this information that's up the top, right? But we've answered A, we've answered B, we're looking at question C, okay? It says, at which stage in the animal's development are stem cells most abundant, right? So what we've got to do is look at the table first. Does this table tell us where stem cells are most abundant? No, the table doesn't tell us. So what else have we got? This little bit of chit-chat at the top. So have a wee look at the chit-chat at the top, right? So see if we look at this, it says stem cells are unspecialised cells, which can divide to make a wide range of other cell types. The embryo contains the highest proportion of stem cells. There we go. That's where they're most abundant. It's in the embryo. So that there, last question, embryo. So again, we're moving on, and this is our transpiration question. Question 11, okay? The rate of transpiration in plant cells can be measured using the apparatus shown below. As the plant transpires, coloured water is drawn up the glass tube. So look at this, coloured water is drawn up that glass tube, and its volume is measured over a set period of time to give the rate of transpiration. Changes in the surrounding environment can have an effect on the rate of transpiration. Select one of the environmental changes listed below, right, by circling it, and look at the question. State the effect of this change on the rate. So if I pick an increase in humidity, what's that going to do to the respiration? Always think about clothes drying, right? So the more humid it is, the more water that's in the air. Are your clothes going to dry faster? No. This is going to decrease transpiration. An increase in temperature. What will this do? Clothes drying? Will my clothes dry faster? Yes. This is going to increase transpiration. Increase wind speed. Are my clothes going to dry faster? Yes. Increase wind speed. Okay. So, suggest how the apparatus could be adjusted to ensure that water is only lost through transpiration. So look at this. I want to know that this water, so let me just change my colour of pen. I need to know that this water is just going up this little glass tube, right? And that none of it is like, look, evaporating out of here. So what would I do? Put in a layer of oil. If I put a layer of oil on the top, it's going to mean that no water can evaporate. So again, still my question 12, the graph below shows transpiration rates of 2 plants, P and Q, okay? So it says, with reference to the number of stomata, suggest a reason for the different transpiration rates of P and Q, okay? So what we want to do is look at this, P, is it transpiring? So is the transpiration more or less? It's got more transpiration, right? Q has got less transpiration. So this question says, in reference to the number of stomata. So one of them is going to have more stomata, one of them is going to have less stomata. So who'll have more stomata? The one that's losing the more water. See the less transpiration, the less stomata, it's losing less water out of the stomata. So Q has more stomata. Oh, sorry, Q, look at that. P. So there's my P right up at the top there. P has more stomata than Q. Or I could do that the other way around. Q has less stomata than P. Name the type of cells which control the opening and closing of the stomata. So what are those cells that either the stomata will be open or closed? Guard cells. So next question, right? Diagram represents a section through a leaf, and it's asking us to name tissue W, right? So tissue W, have a wee look at this, and know that they are mesophyll cells. So what mesophyll cells are they? Palisade, right? Look at this. There's the epidermis. And then look in here, look at this. That's the spongy mesophyll. So tissue W, the palisade mesophyll cells. Okay. Next question. The cells in tissue W have a greater number of chloroplasts than the other cells, right? Suggest the advantage of these cells to being located near the upper leaf, right? So why do I think that being closer to the top of the leaf is going to be better for having more chloroplasts, right? So what you've got to think is, well, what do chloroplasts do? They absorb something. What do they absorb? They absorb light. So if I have more chloroplasts in these cells, what are they going to be able to do? They're going to be able to absorb more light. Okay, we have to have that word more in, so we're going to absorb more light. The leaf vein consists of xylem and phloem. So all we've got to do here is it says pick one of them and describe a structural feature. So if we go from xylem, what's a structural feature? It has got rings of lignin, right? What else could I say? I could say it's hollow, right? Phloem, what can I say about that? I can say it's got a companion cell. I can say it's got a sieve plate. Sieve plates can also be called end walls. And I also have a sieve tube. They're my structural features, right? The xylem are dead, the phloem are alive, but those are not structural features, okay? So there are my structural features of both. Question 13. So the diagram represents the second stage of photosynthesis. And what I've got to do is to name what's X and what's Y. So look, here's the ATP and hydrogen. This is coming from the first stage. So what is coming in brand new here, substance X, what gas is coming in? Carbon dioxide. So that substance X is CO2. Now look, I make the sugar, that's the glucose, right? Storage. How do I store the glucose? I store it as starch, right? See if they did it for cell walls, then it would be cellulose, right? But storage is starch. Here's our limiting factors graph, okay? So what we've got is we've got carbon dioxide concentration along the bottom, we've got rate of photosynthesis up the side. So X, right, what we've got to think is we've just got to think about our rules, right? Now the rules of this is at any point on this line going up, right? So if it's on the line going up, where do I look? I look down. Whatever is on this x-axis, that is what's limiting if it's on the way up. So look at X. Is it on the way up? Yes, it is. So I look down, it's carbon dioxide concentration, okay? Now let's look at the other one, right? So Y, when it's levelled off, I know it's not this thing that's on the bottom because I'm increasing CO2 and it's not having any impact on the rate of photosynthesis. So when it's levelled off, what I want to do is I look up and then I think, see on my line, what's lower on my line here? So what's lower on this bottom line? Temperature. Some carbon compounds found in plants are shown in the list below. It says, so I've got my wee list, carbon dioxide, cellulose, glucose, starch. We need to pick out of this list, right, it's saying raw material for photosynthesis. So what one of this list up at the top do I need for photosynthesis, right? So let's just go through one at a time. Do I need carbon dioxide for photosynthesis? Yes, I do. Cellulose, no, because I end up making that. Glucose, I make that. Starch, I can make that, right? What I'm doing is I make glucose, I can then turn the glucose into starch for storage, or I can turn the glucose into cellulose for cell walls. So what's the raw material for photosynthesis? Carbon dioxide, right? Respiratory substrate. So respiratory, that's respiration. Substrate is something you start with. So think about an enzyme. an enzyme works on a substrate. It's the thing you're starting with. So do I need CO2 for respiration? No. Do I need cellulose for respiration? No. Do I need glucose? Yes, I do. That's your glucose. I break down glucose in glycolysis. Now, here what we've got is these 4 test tubes, right? First picture, look at this. I've got a black cover over my leaf, right? So I need to decide if this leaf is covered in black paper, can it do photosynthesis? Can it do respiration? Can it do both? Or can it do neither? So first thing that we want to think is, can it do photosynthesis? Can it? It's got a black cover over it. What do I need for photosynthesis? I need light. I can't do photosynthesis. Can I do respiration? Do I need light for respiration? No. So it's going to be able to do respiration. Test tube 2, let's have a look. It's not saying anything, look. I've got the leaf. It's not been messed with in any way. So can that leaf do photosynthesis? Yeah. Can it do respiration? Yeah, it can do both. Look at this leaf. It's been boiled. What's happening to enzymes when they're boiled? Denatured, active site changes shape. Both respiration and photosynthesis are enzyme controlled. Test tube 3, neither. Now look at test tube 4. It says it's a variegated leaf, which means it's got green and white bits, but nothing else has happened. So can this leaf do photosynthesis? The green bits, can they do photosynthesis? Yeah, they can. Is anything going to be mucking with respiration? No, it's not. They'll do both. So that one is both. Okay, so our river was sampled at 5 sites as shown in the diagram, right? So let's look at this wee river. Look at the arrow direction of flow. So the river is flowing this way, okay? And what we've got here is, look, sewage. The sewage is going in at point 2. So number 1, I don't have the sewage, but after number 2, I do. Let's look at the tables. The following tables shows the results of analysing the samples at each site. So site 1, look at this. That's pre the sewage. I've got lots of oxygen, less bacteria. But then see at site 2, look at this. That's when the sewage has gone in. I've got the least oxygen and look, the most bacteria. As I'm getting further away from the sewage, the oxygen's increasing. So look, as I'm getting further away from the sewage, the bacteria is decreasing. Okay? Let's have a wee look at table 2. Table 2, organisms present, and we've got our sites. So at site 1, what have I got a lot of, right? So I've got mayfly nymphs, stonefly nymphs, caddisfly larvae, I've got a couple of water shrimp, but then I've got way less bloodworms and sludgeworms, right? Let's compare that to site 2, because site 1 is no sewage, site 2 is the sewage. Look at this. None of these ones now, but I've got bloodworms and sludgeworms, okay? Using data from table 1, describe the relationship between the number of bacteria and the oxygen levels in the water. So look at this first, right? What we want to do is a relationship you do as the. So as the oxygen levels, right? What's happening to the oxygen levels? The oxygen levels are decreasing. So look at this, 1.2 to 0.04. The oxygen levels decrease, right? Look at the number of bacteria. Number of bacteria are increasing. But now look, from 0.4, what's happening to the oxygen concentration? It's now increasing. What's happening to the bacteria? Decreasing. So as oxygen levels decrease from 1.2 to 0.04 units. Number of bacteria. What's my number of bacteria doing? They are increasing from 500 to 150,000. And I put in my wee units per 100 mils. Now that's the first bit. So what do I do now? I need to do the next bit. So I now go as the oxygen I'm not decreasing this time, look, I'm increasing. As the oxygen increases from 0.04 to, and I'm going, look, all the way up to 1.12. What's happening to the number of bacteria? Look, number of bacteria decrease. Now, what are they decreasing from? That 150,000 to 1,250. And again there, what I'll do is I'll just squeeze in my units. So that's per 100 mils. And up here, this is units. Okay, as long as I do the units once, I don't have to do them in both sentences. So as long as I do them in the first sentence, I don't have to re-put the units in the second sentence. Okay. Part 2, it says methylene blue is a chemical which can be used to compare oxygen levels in the water. The lower the oxygen level, the faster the methylene blue changes from blue to colourless. A sample of water from each of the 5 sites was tested. Predict what a sample, right, would lose its blue colour the fastest, right? So let's go back to the start. The methylene blue is a chemical which can be used to compare oxygen levels in the water. The lower the oxygen level, the faster the methylene blue changes colour, right? So the faster it changes from blue to colourless, right? Sample of water from each site. Predict which sample would lose its colour fastest. So I've got to look for the lowest oxygen concentration. Lowest oxygen concentration is site number 2. Next, use data from tables 1 and 2 to answer the following questions, right? Part 1. State which of the organisms in the samples would be found in the areas of high oxygen. So what I've got to do here is just look for the high oxygen. So 1.2, that's site 1, high oxygen. 5, that's high oxygen. So let's look at site 1 and site 5 in table 2. So what am I going to find? I'm going to find mayfly nymphs. I'm going to find stonefly nymphs. I'm going to find the last one. Look at this last one. I'm going to find caddisfly larvae. Okay. So there are my 3 possible options. Okay. The next question here. Sewage in the river is a form of water pollution. Describe the effect this pollution has on the number of different types of organisms in this river. So what am I doing here, right? The more sewage I have, so the sewage in the river is a form of water pollution. Describe the effect this pollution has on the number of different types of organisms in this. I have fewer types of organisms. I have less types of organisms. Okay, it's the types there that's important. It's not just the fact that I've got less organisms, I've got less types of organisms. Okay, bottom one here. Some species are known as indicator species. Explain what is meant by the term indicator species. Now this is just straightforward knowledge. Okay, indicator species is a species which, if is present, or sometimes it's not present, sometimes it's absent, it gives us information about the level of pollution. So your species that if it's either present or absent, it'll indicate for us, it'll tell us about the level of pollution. Either it'll tell us there's more pollution or less pollution. Gardener treated the soil in the area where he planted vegetables with a chemical to increase yield. The chemical added to the soil by the gardener contained nitrates. Give the general name for this type of chemical. So what does my farmer spray on his crops? Fertilizers. Describe the use that plants make of nitrates. So what's the use that plants make of nitrates? To build protein, to make protein. That's my use of my nitrates. Question 15. So the diagram below shows part of a food web. Using information from the food web, answer the following questions. So first one here, A part I, it says identify an organism which is both predator and prey. Okay, so what I've got to think here is I've got to think it's eating something, right? But something is also eating it, right? So the green plants, right? They're being eaten. Okay, it's not predator or prey. See the insects, look at this. Is the insect a predator of the plants? Is it having to like pure sneak up on the plants, jump out and eat it? No, right? So see this. right? Blue tip, let's go further up. Is the blue tip a predator? Yeah, it is. Look, it's a predator of the insects. Is the blue tip prey? Yeah, it is. It's predator of, it's prey of the sparrowhawk. What's my other option, right? So look at the wren. Again, this could also be the wren, right? Because look, it's the predator of the insects, but it's the prey of the sparrowhawks. Last one, the thrush. And again, it's the same chat. It's the predator of the snails, but it's the prey of the sparrowhawks. Part 2. Pesticides are known to run off from the land into rivers and enter the food chains. Name the organism which would accumulate the greatest concentration of pesticides over the period of time. Now, the pesticide gets passed on at every single step, all of it. So this is called bioaccumulation. Who's going to end up with the most? The sparrowhawk. Because at every single step it's getting passed on. State one way in which energy can be lost between stages in a food chain. So I'm losing energy, movement. If I use it for movement, it's gone, it's out of the food chain. Whoever eats me can't get that, right? Undigested material. Feces, I could say, right? See this undigested material again. The undigested material is essentially in the jobbies. So can whoever eats me get that energy? No, it can't. Again, lost. The last one is heat energy. So if I lose it as heat energy, it's gone, it's out of the food chain. So there are my 3 options. Same food web, but another wee question here. It says predict the effect on the population of snails. So we're paying attention to the snails, right? If there was a decline in sparrowhawk, so let's cross out the sparrowhawk, all right? So I've not got sparrowhawk, and what I want to find out is what's going to happen to the snails, okay? So what's a link between the sparrowhawk and the snail? It's the thrush, look. If I take the sparrowhawk out, what's going to happen to the thrush? Thrush are going to increase. So what's going to happen to the snails? The snails are going to decrease. More thrush to eat them. Or you could think thrush, eat more snails. Either one. Again, same food web. Give the name of 2 organisms who are in competition with each other for food. So what I'm looking for is I'm looking for a source of food and it's going to different things because these 2 things are then in competition with each other. So let's look at the green plants, right? Who's eating the green plants? Insects and snails. They're in competition with each other, okay? Now, again, I want to look for a food source. So snails are getting eaten by thrush. Is there anything that's competing with the thrush for the snails? No. So let's look at the insects over here. Who's competing for the insects? Blue tip and wren. Are the sparrowhawks competing with any other species? No. The sparrowhawks are eating the blue tits, the wrens, the thrush. So here's my 2 organisms, insects and snails, or blue tit and wren. So on to question 16, sampling. Okay, so sampling techniques can be used to estimate the abundance of plants and animals. In an investigation into ground-living animals in a woodland, a group of students collected and counted animals they found. Name a sampling technique which could be used to collect ground-living organisms. So what do I dig a wee hole and I set up a pitfall trap. Right, that's my ground-living organisms. So here's my wee bit of my experiment that they've done, right? The students sorted the animals into male and female, counted them and recorded the results in a bar graph. So I've got the woodlice, male and female, the springtails, beetles and centipedes, okay? Up the side of the graph there, I've got a number of individuals. Identify the animal which had the greatest overall abundance, right? So what I've got to do here is I've just got to count them up. So look at this. There's 5 females and there's 3 males for the woodlice. So what's that giving me? 8, right? The springtails, there's 5 and 4. So what's that giving me? 9, right? So far, it's not the woodlice, right? The beetle, I've got 5 plus 2, 7. So Snow the beetle. Centipede, I've got 3 plus 2 gives me 5. Who's the best? Not it's the springtails with 9. So this, springtail. The students concluded that the males were always more abundant than females. Identify the animal for which this is not true. So where is it where you've got more females than males? Sorry. So the only one where that's not true is the woodlice. It was decided that the samples were not fully representative of the area. Suggest how this investigation could be improved, right? If it's not representative, then maybe I've only set a few traps. So set more traps. I could also think, well, maybe some of the stuff has got eaten. So check more often. They're my two options for improvements. So, the distribution of organisms may be affected by abiotic factors. The table shows the results of a study into the effect of soil moisture levels on the distribution of 3 species of plant. So what I've got here is sample sites and I've got soil moisture. Look at what's happening to the soil moisture. The soil moisture is increasing. What I've then got is these number of plants and I've got my different species. So let's look at what's happening to species E. Look at that. As the moisture increases, species E is increasing. Look at F. As the moisture increases, is there really a pattern here? Look, 15 to 14 to 16 to 17 to 13, back to 15. These are all much of a muchness. Look at G, 12, 11, 10, 15, 12. Again, these are kind of all over the place. State which species has its distribution most affected by the soil moisture. Well, species F and species G, do I really care? It's E. Where I've got less moisture, I've got less of species E. Where I've got more moisture, I've got more of species E. Okay. Calculate the average number of plants per sample site for species F. Okay. So all I need to do is add all the numbers up. So 15 plus 14 plus 16 plus 17 plus 13 plus 15. So add all them up and then divide it by how many samples have I got? 6. The answer will give you 15. You could pause, pop it into your calculator. So, our last question here, right? A study found that exercise can reduce the risk of developing some types of cancer. Researchers examined the level of exercise of 1.4 million people over a 11-year period. They recorded any diagnosis of cancer and when it occurred. Participants were asked to keep their own record of the frequency and intensity of their exercise. Those participants who exercised were found to have a reduced risk of developing some types of cancer. These results are shown in the table. Okay, so what I have here is the type of cancer. I've got lung, kidney, stomach, myeloma, bladder, and I've got this average risk of reduction. Down here, it says the study did not take into account factors such as diet and smoking, and these may have affected the results. The fact that participants were asked to record their own exercise is another limitation of this study. The doctor in charge of the research suggested that these results support the promotion of exercise as a means of reducing the risk of cancer. However, it was suggested by a different researcher that further studies would need to be carried out before the results could be considered valid. So let's have a wee look at our questions, right? We look at A. Most research starts off with a question. For example, is enzyme activity affected by pH? Suggest a question that could have led to the research being described. So when we look at this, right, we've got a study found that exercise can reduce the risk of developing some types of cancer, right? So we need to think of the question that they asked themselves. So this study is looking at if exercise reduces the risk of cancer. So what's my question? Does exercise reduce risk of cancer? That's my question. Name the type of graph that should be used to present the results. Now I've got two options here, I could have a line graph, or I could have a bar graph. Which one of these would it be? Look at this, I've got words along the bottom here. It would be this, a bar graph. C. Suggest one reason why the participants recording their own exercise is described as a limitation. So it says that the participants were asked to record their own exercise, and they were saying this could be a limitation. So what could the people be saying, right? They could be outright lying about their exercise. They could exaggerate, right? So they could say, oh yeah, I did loads of running that day. Maybe they forgot, right? Maybe they did some exercise and they forgot about it, right? Essentially what's happening here is they might not tell the truth, right? So they may not tell the truth about this. They may say that they exercised when they didn't. They may say they exercised more than they did, or they might have forgotten and not actually said that they exercised when they did. Last question here, D. A dependent variable is what scientists measure or observe. So what we've got to do here is it's saying identify the dependent variable. So the dependent variable is what the scientists are measuring or observing. So look at this table up here. Type of cancer, average risk reduction. What are they measuring? They're measuring the average risk reduction. So look at this. What's the dependent variable? Average risk reduction. That's what I'm measuring. Okay.